I realise the title might be a little bit different than the one I submitted, but it's the same stuff, just under a different name. So thank you very much to the organisers for giving me this opportunity to speak to you all today. I had a really great day yesterday listening to all the talks, and I'm sure that will continue across the workshop. So I work in this place here, so it's the Living Systems Institute at the University of Exeter. So you might take from that we're going to take a slightly biological bent in my talk this morning, although you don't need to know very much about neurons, and I'll tell you everything that you do need to know. So we'll keep the biology quite minimal. What I do want to talk about at the beginning, though, is just to give some motivation for the kind of things that I'm looking at. So in particular, the bumps and waves that I mentioned in my abstract. A very quick review of single neuron behavior to motivate moving up to the network level, and then finally getting to the bulk of what I'm going to talk about, which will be about spiking neural field models. So this is just a picture that I guess almost everybody who works in sort of applied maths or sort of multi-scale phenomena has some variant of. Here's my neuroscience one. So this is just to show you a little bit about the kind of scales that I think about and where I think I work. So here is sort of the full brain. We have sort of brain regions, sort of local microcircuits, individual cells, and the proteins that form ion channels that kind of govern behavior. I typically sit somewhere between here and here. Actually, although put it here, I'm probably more talking about cells, circuits, and sort of very small regions today. So the idea is to try and couple behavior at single cell level to waves and bumps, so different types of solutions you might expect to see at a tissue level. So I'm going to start off with some psychophysics. So this is not even really, well, I suppose it is true biology because there's some experimental recordings here. So these are not experiments that I do. But the idea is that you're taken into a little booth and you're shown a whole bunch of different shapes with a whole bunch of different colours, and you're asked to remember some feature about the things that you're seeing. So, for example, you might be shown a whole bunch of shapes, and some of them might be green triangles, some of them might be red circles. You're asked to remember that, and they say, okay, we're going to take these shapes away from you, and then half a second later or something, we're going to show you a whole bunch of shapes, and then you need to select all of the shapes with the feature that you've been asked to memorize. So here's an example where the participants have been asked to recall red. So after the delay period, they'll be shown a whole bunch of shapes, and they have to select all of the red ones in some period of time. And what you see if you do that, and I think these are monkey data actually, not human data, but if you look at cells in the visual cortex, you find that they encode for these different kind of percepts. So there'll be cells that encode for triangles, cells that encode for circles, and cells that encode for colours, green and red. And if you look at the activity, so firing rate here, so spikes per second is taken as sort of a functional readout of what the cells and the cells in the network are doing. For the cells that are encoding features that are not being asked to memorize, they sort of maintain this basal level of firing, right? So this is kind of normal cells, kind of fire quite sparsely, so around 9 spikes per second is not super high. Conversely, if you look at the cells encoding for the red percept, in the delay period, so here is the stimulus, there's a delay period here where nothing is being shown, and then the M stands for match, where they're being asked to match the things that they've been asked to memorize. You see an increase, a persistent and maintained increase in firing rates over the red cells, which then drops once they start doing the task. So when people talk about things like working memory, this is a really good example of something that they're being asked to store. So the stimulus has been taken away from here at this point, remember. So this is just persistent, sustained firing in these cells encoding for this percept. Bless you. The thing gets even more interesting when you think about stimuli that are now continuous. So these were categorical stimuli. Now we'll think about something that's continuous and in particular imposed on a ring. So these are diffusion gratings that lots of people in psychophysics like to use as a prototypical stimulus. So this grating will actually be moving. I'm not putting a little movie here, but imagine this whole grating is moving in this kind of northeasterly direction here. Or actually no, the other way around, it's moving southwest. And so again, if you play the same kind of game, so you show them this stimulus, and then you say, okay, there's going to be a delay period now where the stimulus is removed, and then they'll be shown a whole bunch of gratings, and now select the ones that were close to the original stimulus percept. And so here you can see cells that are encoding for a particular orientation, so 225 degrees. Again, they show the same persistent and sustained increase in firing. Moreover, if you actually look at cells in the visual cortex, you find there's a nice topographic mapping. So actually, that cells that encode for angles that are close to each other will also be geographically close to each other in the visual cortex. Okay, so there is this nice mapping. And so what that means is if you look at a sort of section across the visual cortex and you just scan over and you show these kind of stimuli, what you find is a sort of fall off of firing rates from the actual preferred direction, which is being shown, and it falls off like some sort of bell shape. Okay, so I'm going to refer to this thing here as a bump, because it looks like a little bump of activity, where we have maximal firing rates at the preferred orientation, and then it falls off with some rate here. Okay, so those are bumps. I'll refer to bumps quite a few times over the talk. I want to motivate one other thing, which again comes from a psychophysics experiment. So this is a kind of funky experiment where you're sitting there, you've got essentially cameras shining into both eyes, but the important thing here is that each eye is being shown a different stimulus. So here is an example where the left eye is being shown this red grating, which is sort of angled southwest-northeast, and the right eye is being shown a blue grating, which is shifted by 90 degrees. And if you ask people what they report when they go through these experiments, what they say is, well, okay, sometimes I see the red thing, then it switches to the blue thing, and then it switches back, and then switches back, and it keeps doing this. Okay, it's not a periodic switching. There's some, it's a stochastic process, but there are certain kind of reproducible phenomena. So that if you increase the salience of one of these percepts, actually it's the other percept that you see less often, right? So the dwell time in that other percept will go down.
Is it statistics of such priming in binocular rivalry is known?
There's
how much you can prime for this and quantitatively?
So that's a good question, and it's ongoing. So the things I was talking about increasing salience, and you can have predictable effects on statistics, these are all known as Lebel's propositions. So they've been investigated in humans and in monkeys. I think they're the main two. So they are statistically reproducible. Not a lot is known about exactly where it comes from. So this is why it's kind of psychophysics. We observe this quite macroscopically. There are some, I'd say, pseudo-mechanistic mathematical models that help understand where the switching has come from. So sort of winnerless competition models where you're switching backwards and forwards. And they can be fit to the data quite well. To my eye, they fall short of being truly mechanistic because they don't really explain where it comes from, but at least we can match the statistics. Okay, and so if you play the same kind of game that we did for the delayed match to sample task, and you have a look at what brain activity looks like, so this now is in human. So they're measuring something here called the bold signal, which the long of the short of it, it is a readout for functional brain activity. So this is a different kind of percept, but it's the same kind of idea. The left eye is being shown these kind of spiral things, the right eye is being shown this sort of radial arms with low contrast. And if you look at the brain activity during the switches between these percepts, you find evidence of waves moving across the visual cortex. Okay, so you can see here, so this is activity in this blue region here, and activity in this red region here, and you can see that the latency to the red region is slightly shorter than the blue region, and actually if you look at this across lots of different stimuli and lots of different regions, you find evidence of propagation of waves. Okay, so somehow bumps are really important for encoding working memory, waves are really important for switching percepts. Okay, and I don't think I'm going to say too much more about these psychophysics experiments. The point is, bumps and waves are important for computation, are important for perception.
How do we interpret these waves? Are they electrical signals?
Yeah. Well, yes and no. So they are. So the bold signal here is essentially measuring oxygenation level in the brain, and the idea is that that is supposed to be a caveat for energy demand by brain regions, which in turn is motivated by the fact that it has to maintain electrical activity. So if this signal is not a direct readout of electrical activity, but it is a surrogate for it. Okay, so now let's talk about how we might approach this kind of problem using very, very simple mathematical models. So we're imagining then I've got a whole bunch of neurons that are connected in a ring. I'm just showing you connections between one neuron here, but effectively this is copied. So the whole ring is isotropic and it's all symmetric. And in these kind of systems, you can generate behavior that kind of looks like this. So here is a continuum version of this, where now I'm just packing loads of neurons in. And what I do is I say, well, locally, if I don't push the cells, they just decay to some resting state, which for me is just going to be 0, right? I can just scale this however I like. And then I say, okay, well, in neuronal networks, they tend to talk to each other, and they tend to talk to each other over actually quite long distances. So what I get here is a coupling kernel that filters the activity at some neuron, feeds it through some sort of temporal filter. So I call this a firing rate function. Some people call it a transfer function. It's just some filter of if the neuron is firing this fast, this is what I feel as an input to me. And then we have a distance-dependent kernel that describes how strongly the neurons talk to each other. And so the idea here is that there's some dependence on exactly how far away the neurons are. And this is relatively reproducible. It's taken from data going all the way back to Vernon Mountcastle in probably, when was that, 1910s? So this is distance dependent, this is a temporal filter, and in these kind of models you can produce activities that look like this. So this thing here it looks like my bump, so I'm going to call it a bump. There's some threshold level of activity here, so H is some threshold level of activity. So these cells here are somehow on, they're firing, they're electrically active. Cells below this threshold are not, and so all of the recurrent activity in this network is being generated and sustained by the stuff in the blue region. Okay, so how do you analyze that? Well, it turns out if you make some approximations, it's pretty straightforward to do this. So this firing rate function here is very often taken to be a sigmoid because we think, okay, as we drive these cells, they come on, they saturate, and so we can approximate that with a sigmoid. If we take the high gain limit of that sigmoid, we can just replace the firing rate with a heavy side. So this is now just a step function which steps up at h. From there, we can say, okay, well, which bit of my tissue, right, so this is a one-dimensional tissue, which bit of my tissue is actually active in this bump state? So I define that by my set BH here. And then my equation here, I can collapse all of that down to just something that looks like this, where I'm just integrating over the distance-dependent kernel over my activity set. So that's really nice. I can actually just solve that at only 2 points, so this point here and this point here. And in actual fact, because the whole thing is symmetric, it's equivalent to do that at just 1 point. So I've now just got one equation that I can solve for understanding where this profile is at threshold, and I can reconstruct the entire profile. Okay, so somehow I've mapped this whole thing now down to just finding a solution to a one-dimensional system. Moreover, if I do my linear stability analysis of this, so I do the usual thing of putting in some small perturbation, I can get an eigenvalue problem that, again, because of the properties of the Heaviside function, I just have to care about perturbations at 2 points, one here and one here, and that will deal with things like translation, shrinking, and growing of the bump. Okay, and so this is all very, very nice, and it basically collapses this big problem down into something quite simple. I'm not going to talk too much more about this kind of neural field model. I think Rachel Nix might talk a little bit more about these types of models later in the week. Okay, so I said before, I want to do something coupling single cell behavior to these bumps and waves. And I haven't really talked much about single cells. I've just said there are some cells that have some activity and I can couple them. So here is a little bit more biology. We won't go through too much more. So for those of you who haven't seen these kind of experiments, and now going back to your question about electrical activity, this is direct electrical activity. So here is a neuron that I just grow in a dish. And you can basically stick a pipette with an electrode in it. And if you suck hard enough, you can rupture the cell membrane, and now your electrode is inside the cell. If you have some other electrode outside the cell, you can now measure the voltage across the cell membrane. So that's what this is here. And this is actually, the cell is not being stimulated here. So this is just kind of spontaneous activity in the cell. And then you can do all sorts of things with it. You know, you can give time-varying stimulation profiles or whatever you like. But the point is, this is what single-cell behavior actually looks like when you drill down, you put an electrode on a cell. So here we can see sort of the main thing is these very large voltage deflections here, which we call action potentials, or colloquially spikes because they're spiky. So this is the kind of stuff that I'm interested in. Okay, so how do we describe that? Well, we have some great work by Hodgkin and Huxley that was done back in the late 40s and early 50s. We have them to thank for sort of the prototypical model that most of us, when we first think about what a neural activity looks like and how it can be described, we would go to something like this description to start. So the idea here is that, well, we're measuring the voltage across the cell membrane. The cell membrane acts a bit like a capacitor. There are Nernst potentials because the cell is maintaining electrochemical gradients across its membrane. And there are proteins that basically open and close. And when they're open, they allow ions to flow through, and hence current flows. And when they close, they don't. So these are variable resistors. And the point was, by mapping this cell activity to this sort of electrical circuit, which we know how to write down equations for this, they wrote down some nice equations that basically capture the broad features of neuronal behavior. And most of the kind of models that we use, which are sort of more mechanistic in nature, are some variation of this with different kinds of bells and whistles on. Here is a simulation of this model. Okay, the real data look pretty noisy, and in fact they are. They don't look like that, but the point is, qualitatively, the features are well captured. Okay, so let's just take a step back, because I mentioned something about thresholds before, so I want to reintroduce those. So here is actually a restricted version of that Hodgkin-Huxley model, now down to just 2 equations, so we can look at things in the phase plane. And so some of you will probably know this already or have seen something like this, but for those of you who haven't, just introducing the notion here of excitability. So for neuroscientists and others, cell excitability is this notion that if I drive the cell a little bit and I let go, it'll just go back to baseline. If I drive it a bit harder, it will still go back to baseline because here it's a globally attracting fixed point, but it will do so by making a really big excursion. And this big excursion is the same as an action potential. It's a spike. Okay. So what that means is that somewhere between this point and this point, there is a manifold that separates things that go straight back to fixed point and things that first spike and then go back. Okay. So this, somewhere in here, is my threshold, and those are the kind of things that I want to understand, because once the cell has made a decision to spike, actually that behavior is pretty repeatable, prototypical. Actually, the spike shapes don't change very much from spike to spike. It's really just this decision, spike or no spike. So we call this an all or nothing response. Okay, that model is still too complicated in general for us to do anything nice mathematically with. So I'm going to go back actually to a much earlier model. So Lepic was building models not unlike the one that Hodgkin and Huxley developed, but it was simpler in some sense and more complicated in others. He was experimenting on frog's legs, so doing the same kind of thing, so putting electrodes in, pushing stimulus through until he got a response, which you can now measure because the frog's legs will twitch. And he came up with a model that looked like this. So here, the resistance here is no longer variable. Okay, so it's really just a simple integrator. It's just integrating information. But what he did do is he said, well, I'm going to assume that there's some threshold here. Once the cell voltage reaches that threshold, I spike. Because he said, well, look, I mean, the spikes kind of are the same all the time, right? So we just need to care about the spike timing, not so much about the actual shape itself. So he created this model, and later this model was referred to as the integrate and fire model, and that name kind of stuck. So this is the other sort of big class of models that people who look at sort of single cell electrophysiology work with. So they're either using these conductance-based models from Hodgkin and Huxley, or they're using these much simpler integrate and fire models, although there is one caveat with that, is that now we have some non-smooth dynamics given the interaction of the cell voltage with this threshold. Okay, so this is the model that I'm going to work with today. I'm going to work with the integrate and fire model. And so we can reproduce the same kind of network that we had before, where we had this sort of ring topology. So again, we have some decay back to some baseline state in the absence of any coupling. We have this reset thing to deal with now, where when the voltage hits some threshold, it just gets mapped down somewhere else to some lower voltage. And that's kind of trying to imbue the cell with some sort of refractoriness, right? So it has to take some time again to build up some activity before it can spike. And then we have this coupling kernel. So alpha here is my temporal filter. So it's the temporal kernel. Again, it tells me if a cell spikes and I'm connected to that cell, what do I actually experience? And then some wij here. So wij is just sampling from this weight-dependent kernel I mentioned before. Okay, so again, there's this distance-dependent feature by which cells are coupled. So this is just to remind ourselves that we're doing this hard reset at threshold. We have cells that are reciprocally coupled, and this is the kind of coupling kernel I'm working with. So again, motivated by dynamics in the visual cortex. So cells that are close to each other tend to make each other more likely to fire, so that we call them excitatory. Cells that are far away tend to suppress activity. So we have what's called a lateral inhibition kind of kernel. To make my life easy, and again, to recapture what we were kind of looking at before, I'm going to take some continuum approximation of this. So all I'm doing is I'm mapping this to this thing here, and now my sum here gets replaced with an integral. This sum here is summing over the fact that a single cell could spike multiple times. Okay, so that's all this sum is really doing. So this is what I'm going to refer to as the spiking neural field model. Okay, so I mentioned at the beginning that I care about bumps and waves, so I better check that this model actually does produce bumps and waves. So this thing over here on the left is a raster plot. So space is this way, time is this way. Every time the cell reaches threshold, I put a little dot. And so because I only care about timing of events, this sort of gives me a good overview, a summary of what the network is doing. Over here, I'm also plotting the synaptic kernel. So the idea here is that each cell is feeling some term that looks like this, which is the sum of all of the inputs it's receiving from the network. So I'm just going to plot that because that tells me what's driving network activity. So in some sense, we can think about these neurons as being a nonlinear filter that takes this input and spits out spikes at various times. So that's why I'm plotting these two things here. My beta parameter, which is a parameter for my temporal filter, is telling me how quickly I'm integrating information from other cells. So typically, when a cell spikes, it will send out a signal to other cells that goes up and it comes down. Beta is controlling, for me, the rate at which it comes down. So how long that signal persists over time. So for low values of beta, I get this very, very nice bump. Right? So if I were to look, for example, if I were to average across time and have a look at just that across space, I'd recover the kind of bump that I showed you before, both from the psychophysics experiment and from the Amari neural field model. If I increase my beta, so now that decay rate is a little bit faster, I still see a bump, right? I've still got something that's localized in space, but now it's not pinned to where it was initiated. Okay, it just kind of wanders around now, and this wandering will continue. Certainly for as long as my simulations ran. And to generate these things here, I just stimulate the network a little bit, some random initial conditions. I just stimulate it a little bit in the center of my field, and I get these reproducible results. I also, if I initialize my system slightly differently, and I use a slightly different value of beta, I have something that starts off like a bump, but then it then collapses into a wave. Okay, and here the key point here is I want to show you that this system supports lots of different types of waves. So here I've labeled them TW2, TW4, which is just a label that tells you if you've got a packet of spikes that's moving along, the subscript just tells you how many spikes there are. Okay, so it's a pretty simple naming convention. But these simulations were done just by changing initial conditions, but at the same parameter value. Okay, so sometimes we get spikes with bursts with 2 waves, sometimes we get waves with 2 spikes, sometimes we get waves with 4 spikes, and then it motivates the question, well, how many of these things are there? Where are they stable? What's the link between bumps and waves? So that's what I'm going to talk about now. Okay, so this question about bumps and whether or not they have anything to do with waves, so are they connected somehow to the waves or are they different? Right, so we have this evidence from psychophysics experiments that both bumps and waves are important for actually what we perceive and what our brain is doing when we're given some task to do. Are they actually connected in any kind of way? Because of the kind of oscillatory nature of this network, the only actual stationary state that we can have in this system is the all-off state, right? So where none of the network is doing anything. Okay, so that's kind of like when the task is stopped or we're engaging different parts of our brain. So there are no other stationary states that we can see here. So these bumps are not really like that. But we do know that as we take the number of cells in our system, so we're taking something closer and closer to our continuum approximation, we really do get something that looks like a traveling wave. Okay, so here is the sort of the main story, and I'm gonna fill in the details a little bit more as we go as to how we concluded this. So let's focus first on this bifurcation diagram. So I've got my beta parameter here, which is the rate of my synaptic decay. On the y-axis, I'm plotting the width of that packet of wave as it's moving. So we had waves that are moving with some number of spikes. I just take the distance from the first spike and the last spike, and I call that thing delta. And what these things here are, this is the all-off state. So this is a steady state, and this is always stable. This is linearly stable. What I have here then are branches of waves. So this labeling will just continue, right? So this will be a TW5, 6, 7, 8, 9, all the way up to 160. 160 is not a limit, it's just where we stopped computing them. I think ultimately we got up to something like 250. Then it actually gets quite long to do these things. But in principle, this just keeps going on and on and on and on and on. What we find is that for low values of the spike number, these waves are stable. So blue here is stable. And as we increase the spike number in these waves, we start getting instability. So there's points, these are all hop points that are going somewhere down here. They move further and further left until eventually we have branches of waves that are now all unstable. And so what we're thinking here is that, so I haven't filled in all of the details yet. Over here, we see these bumps. So over here we see waves, over here we see bumps. But when we actually look at the transition between going from something, remember we start with some kind of initial condition that starts to produce a bump, and then that bump collapses to a wave. You can also generate things that kind of look like they're doing the opposite. They start with a wave and they collapse into a bump. And so what it looks like is on the way to forming that bump, actually we spend a lot of time bouncing around between unstable waves here that have different numbers of spikes in them. And I'll show you a little bit of numerical evidence for that later. Okay, so that's the summary kind of bifurcation diagram. I should tell you a little bit about how I actually produced, or how we produced this diagram first. So we're not the first people to study these kind of networks. So actually the sort of first numerical studies were done already by Carson, Chow and Carlo Lang, where they were just doing numerical studies of something that looks like this and saying, look, it reproduces the kind of bumps that we see in the visual cortex. So that was kind of our motivation for starting with this. The traveling wave constructions and some numerical results about wave speed were done by Biodermatrout in the late 90s and early 2000s. And the stability of a single wave, or a single spike wave, so just one spike in the packet moving around, was done by Paul Bresloff. And so some other results for periodic wave trains were also done by him later. And more recently, people have started to look at these kinds of systems under additive white noise forcing. There is a question mark as to exactly what the statistics of these bumps wandering around is. So a paper by Zach Kilpatrick and Biodermatrout in 2013 suggested that you can just think about the bump position as essentially being a diffusive process that just moves around. That's been challenged recently, although this paper is still in the archive. It's not yet been published to the best of my knowledge, which suggests that that's not true and that there is some biased motion of these kind of things. Okay, but there was not yet a full theory about how to compute stability of these kind of things. So that's where we kind of entered. Okay, so when we have a system of equations that look like this, remembering that we've got somehow to deal with these reset conditions. Okay, so this whole system now is non-smooth because I keep getting voltages that hit this threshold and get reset. So instead of thinking about it like this, we can think about the system as just an event-driven dynamical system that's just mapping the system state between spike times. And now that's quite nice because we don't have to deal with the kind of non-smooth behavior anymore. We just need to know about when these spike times are. In general, of course, that's a hard problem, but if we restrict ourselves to thinking about traveling waves, that problem becomes much easier because these are very, very predictable. So when we do that, we get a very, very simple now linear equation. So this is defined at a single cell, a single node, and I'm doing all of the coupling between other cells with this variable that I called S. And if my cell, so this is true for all neurons between firing events. So effectively, between firing events, I can just solve all of these systems. It's linear. We can all solve linear differential equations. So that's quite nice and easy. And then when we fire, we just augment we reset the cell that fired and we augment this variable, the synaptic variable here. Okay, so in this integral form that I showed you earlier, in this weak form now, so I've integrated out the whole thing, I can separate the whole system solution into two parts. One that's to do with the fact that I have to keep resetting neurons as they fire, and one that's capturing all of the synaptic input for the cell. Okay, so This is nice because now this just tells me what the solution is. I need to be able to integrate this thing, but if I make some judicious choices for this and this, I can do that. I should say the theory is quite general on what we have to put here, right? It's just that you can do some explicit calculations for specific choices. And the idea here is that what we want to do is we want to specify some properties of these things here. So this tells me when the neuron at position Y reaches threshold, okay, and we are allowing this to happen m times, okay. So we call these the firing functions, and what we want to do is to write down a mapping that takes these firing functions and gives me essentially back my spatiotemporal profile. And if we can do that, then we can perturb the firing functions themselves and examine what happens to the profile and look at stability that way. Okay, so just for convenience, we separate these things out into, so we've got all of these things that are to do with the reset, and we've got these things that are to do with the synapse. So just for convenience, I'm going to separate them. But the point is, they're all things that are picking up these firing functions and returning to me a linear mapping that's going to tell me about my spatiotemporal profile. And then all I need to do is make sure that whatever this thing is here, that when my firing function tells me that a cell at position X should be at threshold, that my voltage is also at threshold, which I've set here to be 1. So threshold is at 1, reset is at 0. Okay, so that then gives me a system of m scalars where I'm just going to say, well, I'm just going to put my origin to be at the front of the wave. Okay, so I'm free to do that because the whole thing is translationally invariant. So I do that, and then I've used a travelling wave ansatz here. So my travelling wave ansatz just says, well, my firing function should look like this. So there should be a common wave speed c because the whole packet is moving along. And then the spike times at a given cell are just going to be offset by some time, capital Tj. Okay, so common c, different Tj. I'm going to set T1 to be 0, and then that gives me a system of m scalars because I assume that there were m spikes in my wave packet. So here there are 5, and I have n equations to do that because I need to enforce. I need to enforce that the system is at threshold at these spike times. Okay, so that's quite nice. Just put that in your favorite root finder. For the wave stability, we do something similar in the sense that what we do is we perturb the firing times. So we give them a small perturbation that I'm going to call phi. We then linearize our mapping, our operator v. So we get this dv here, and we enforce that in the perturbed system, that again, the voltage of the perturbed system has to be at threshold at the perturbed firing times. So that gives us a way of comparing the dynamics around some common point. And when we do that, we can form this dv. Okay, then we can look at the kernel of this thing, and we can extract a spectrum that way. Okay, so that's what we do. And we put in perturbations of this form and its complex conjugate, then we find the kind of spectrum that we're looking for. And typically what we see when we do this perturbation is this dotted line here is the travelling wave. We now perturb it, we extract the eigenmodes corresponding to these eigenvalues, and we see some oscillations around the firing, the actual true travelling wave. Here they're shrinking, so this wave is nice and stable. Here they're growing. You can't really see them so well here, but it is oscillatory here. And then because of the non-smooth nature of the system, if they keep growing and those things then interact with the threshold, then I lose the entire spike here, right? So this thing here will destabilize and this will drop down to being a wave now with just 2 spikes rather than 3. Okay, and then we can compute all of these eigenvalues by root finding. This is part of the reason why I stopped at 160, because although most of this process is automated, you still need to go and find initial guesses for them. So I stopped at some point. And so here's the sort of generic picture that we see along all of these branches. So here I'm just isolating a wave with 3 spikes, as shown here, but the same kind of idea is true for all of these things. So as we go to the right, so as we increase beta, we go through a sequence of hot bifurcations that generate these kind of oscillatory modes that I mentioned before. So here is the spectrum in the nice stable wave profile. Here I've got one pair of eigenvalues that have crossed over into the right-hand complex plane. And then if we look at the other side, we have some kind of strange non-smooth phenomenon. So at this point here that I've labeled G, the wave profile that, remember, has 3 spikes in it, we get a tangential crossing of the profile with the threshold again. Okay, so the system is trying to generate a fourth spike. And if I continue to decrease beta, the whole thing collapses and I get a bump instead. Right? So there's no wave here anymore. I just get this bump kind of thing here. So this really is a termination of the branch. Over here, it's just a destabilization. Here, the branch terminates. And the point is that I have solutions here that are not admissible because I need my voltage profile to remain bounded above by my threshold. Okay, so here's just the picture then showing, I said before, that kind of stuff was the same for all of these branches. So here's showing you what happens. So now on my y-axis, I've got the wave speed rather than delta that I showed before. But the point here, as I move down here, I'm increasing the number of spikes in my wave, and I'm shifting where the Hopf bifurcation is. And at some point, I think for this parameter set, it was something like a wave with 54 spikes. After that, all of these waves are always unstable. Okay, and then this is just showing you now all of these grazing points. So this is what the family of grazers looks like. So this is all the way down to 200 now. And so we thought we'll just have a look a little bit closer at what's going on at these grazing points here. And so we follow these around. So remember, the number of spikes in the wave packet is increasing as I go down this way here. But also the wave speed is going down, right? So these packets are getting more and more spikes and they're moving slower and slower and slower and slower and slower. And if we just look at the scaling properties, so this is some numerics that are looking at the wave speed, c, which is going in this direction, getting slower and slower, and the offset between the first and the last spiking time. Okay, so this is scaling like m, so the number of spikes. And the delta, this width that I was plotting earlier that I said is something to do with the bump width, is the product of these two things, right? So it's c times tm. So then you can see that, okay, this thing scales like 1 over m, this thing scales like m. So if I take this product, ct, m, I get something that asymptotically looks like it's converging to some finite value. Moreover, if we go back and we have a look at the Lang and Chow paper, which did numerical simulations of effectively the same model and showed that it produced something that looks like a bump if you do some time averaging, we said, well, okay, let's just have a look. As we increase m here, just look at the grazing points and work out what the distribution of interspike times is, okay, which is what I'm plotting here for m equals 230. And we found that this thing here lies exactly on top of this black line here, which is the black line from the Lang and Chow paper. So it's some suggestion here that the bump, the thing that they referred to as a bump, that was this time average thing, is well captured by looking at these grazing points and looking at the interspike intervals. Okay, so I said before that we can have a look at solutions that start off bump-like and go into waves, and the converse is also true. So here now is a set of, it's the same bifurcation diagram I showed you earlier. I've also plotted it flipped as well because the waves can go backwards, so they'd have negative wave speed. There's a whole bunch of stuff that's not filled in here because numerically it gets really difficult to do after 200. But what I want to do is just to show you what, if I initialize the system on a wave and just let the thing evolve, what happens. So we'll start at point 3. Here is my initial thing, and eventually it collapses down to this purple region here, which is the wave up here. You can just about see there. On the way to doing that, it visits different other wave states, and the way we had a look at that is we had a look at the profiles, but we also used level set methods to calculate some instantaneous wave speeds. And so that's what I'm plotting here. So in each of these colored regions, I'm plotting an approximation for the wave speed histogram. So this orange thing here suggests that there is a wave speed with this speed here. I've got a blue region here. Okay, the dynamics I've got a little bit of the initial bump still here. But the point is there's some evidence that this solution is visiting something other than this wave here. If I look at point 2 here, so now ultimately I'm going to collapse onto this meandering kind of bump. So it's bump with sort of fixed width, but one that moves around. Again, if I look at the histograms of wave speed, I can see evidence of waves moving in this direction here. So this yellow thing here. Something that's relatively stationary in this red region here, and the initial wave that I tried to put the system on in this blue thing here. And if I go all the way over to the left, so now beta's getting quite low, I think there might be one wave or something that I can land on here, but mostly there are no stable waves here. And now I just go to this kind of fixed position here. So here is just one example of this kind of thing here. So you can see this nice wave here. Eventually it becomes to some bump that has some nice fixed width. I'm going to show you 3 panels that are one here, one here, one here, just to show you what these things look like. Okay, so here you can see clear evidence that the system looks a bit wavy, but we're now with these oscillations and where these firing times are. Remember, these are the raster plots showing you where the neurons are actually firing. As I continue to go up, I generate different kinds of wave profiles where now I'm going to pick up, due to this graze, another threshold crossing that's going to grow, it's going to get added to this here. And this thing continues. And as this is occurring, you can see that the wave speed is dropping as I'm going here. And I'm getting more and more spikes here. But there's still something wave-like that's going on here. Okay, I think in the interest of time, I think I might not go through the other stuff. So we also studied this kind of behavior in a Markov chain model. So this is getting even more abstract, where we're now saying we're not even going to think about integrating information. We're just going to say neurons are spiking, they're quiescent, or they're refractory. You can still find bumps, you can still find waves in these kind of systems, and we've done some analysis of that Markov chain model to understand what it looks like. In the interest of time, because I guess I'm running out of time, right?
Yeah.
So I think I'll stop there, and I will summarize. I'll have to get through this slide. Okay, maybe I'll do it here. So I'll summarize by saying that the interface dynamics approach here, where rather than solving the sort of full spatiotemporal profile, I just care about dynamics at very specific points in my system. So for the Amari model, that was just the 2 points delimiting the bump. For the model I studied, it was where is the system at threshold? Where are these firing time events? The bifurcation structure of the waves that you get out of that is intricate, but again, it can all be understood by looking at these very specific points and the dynamics around them. Moreover, we can actually construct a linearized operator there that tells me something about the wave stability. And so now we have a theory for any different types of waves. And I didn't show you them, but there are also other fun games you can play by saying, okay, well, what happens if this wave bumps into this wave? What happens? Well, sometimes you lose parts of the wave, and sometimes they move as a composite structure. Right, so it looks like, okay, I've got a wave with this many spikes here and a wave with this many spikes here, but they can all be understood using exactly the same kind of framework. And we think that these bumps may be defined as the limiting process, essentially where you start with a wave, you pack more and more spikes into it, and as you do that, the thing slows down until it eventually limits on something that stays static. And we think that this is similar, and I'm not really someone who works in turbulence, I don't know very much about it. But the fact that we can see evidence that our solutions are visiting these unstable waves on the way to this spatiotemporal chaos that defines a bump, we think that this is similar to the transition from laminar flow to turbulent flow in a pipe. And I want to thank my collaborators. So Daniele Avitabile, who I worked with when I was in Nottingham, he's now at VU Amsterdam, and Josh Davis, who is a PhD student who sadly decided to leave academia and now works with DSTL, and the funder. And that's it. Thanks very much.
Thank you, Cal, for a very nice talk. We have time for some questions, both in the room, and if you're online, please type your question into the chat. Yeah.
What sets the width of the bump? And if I may be allowed a second corollary question, do you have any intuitive explanation as to why there is a bump to begin with? I can understand the traveling waves. The bump is a bit more peculiar.
Yeah, so okay. So I do have an answer to the first question. I'll come back to the second question. So the width of the bump is set by a few things. I've studied a really simple version of neuronal dynamics, right, which is just this kind of linear integration of information up to spike. It is really important, the behavior of, I've lost it now, here. this width is really important. So the distance between this bit of the excitatory kernel, so this is the way that the cells are coupled, so this width here. So these lateral inhibition kernels, this distance here is something that is one of the key governing factors of the width of the bump. In fact, if you just study a system where you don't have this lateral inhibition, you just have pure excitation, so spikes are always inducing spikes in other cells, you find that actually it's not constrained, right? So it's not going to form a bump then. You're just going to keep adding spikes and adding spikes and adding spikes until you get to some sort of wave train. The second question is, yeah, I don't really know, to be honest with you. Yeah, you're right, the waves are kind of easy to understand. The bumps are much, much harder to understand. My working hypothesis is that actually and this is coming from actually some of the stuff I didn't have time to show you, is that inside the bump, I've got lots of waves that are forming a little bit like convection cells. I've got some waves that are trying to go this way, some waves that are trying to go that way, and actually overall, because they're all coupled in this same way, somehow that motion is balancing each other out. Of course, instantaneously, some waves are going to be going more to the left at times than others, so that's why we see this meandering. All of that is based on numerical simulation and just looking at things and trying to spot the waves in the solutions, so it may well be that I'm just seeing what I want to see. But I agree, understanding what bumps really are in this system is really complicated. I can't even write down something that says the solution to this problem will define a bump. I hope by the time I finish my career, I have an answer to that question.
Okay, we'll get you back at the end. Any more questions? There don't appear to be any online.
Yeah,
Oh, there you
this
go.
is very much related question. So is the simple, is there an intuitive way to understand why the waves with small numbers of spikes go faster? I mean, you see that very clearly when you had the 3 go to 2, it just immediately accelerated.
That's a good question. So the short answer is I don't exactly know. What I do know is that as the waves are destabilizing, what tends to happen is as the waves are kind of falling off, because the distance between the spikes, the cells that are firing, is getting further and further away, they're getting deeper and deeper into this inhibition zone. But that only tells you transiently what's going on as you lose behavior. It doesn't tell you asymptotically. I'm very confident it is something to do with this inhibition kernel here, because again, if you remove this, you don't see that behavior. The waves with arbitrary numbers of spikes all converge to the same wave speed in that system. So it is to do with the fact that you're picking up more inhibition.
I have a very brief question. So presumably you're considering a very local patch, so you have homogeneity in X and you also have a reflection symmetry.
Yeah.
In the real brain, presumably there's all sorts of other things going on. Do you know how these things interact with inhomogeneity, for example?
So again, it's a very good question, and yeah, you are correct. Actually, I'm using here a very, very simplified version of what this kind of kernel might look like. In actual fact, the system in real brains is even more complicated because you get sort of patchy connections. So I mentioned before about orientation preference, and I said that there's some sort of geographic map. But it's also true that that kind of thing sort of is tiled across the cortex, and you find that cells that have the same kind of orientation are also more likely to be coupled, even if they're really, really far away. So there's an anisotropy in terms of the coupling. In general, I can't talk about the exact kind of waves that I'm talking about here, but the kind of structures looking at bumps and things that are not too dissimilar to Turing kind of patterns, they do survive those kind of things. But the heterogeneity can also add other kind of effects, so you get kind of ringing effects where you get other. There is some interesting theory that covers specific cases of heterogeneity. In general, of course, there's lots of unanswered questions.
Yeah. Okay, that's great. Let's thank Kyle again.